Hello everyone, I am Dr. Akshatha Nayak, Assistant Professor in Biochemistry, Manipal Academy of Higher Education, Manipal. I am here to discuss on a topic called vitamin A metabolism. So, we will discuss the topic under these objectives: chemical nature, coenzyme forms, sources, requirement, biochemical functions, deficiency symptoms, antagonistics of vitamin A. So, as we all know, vitamin A is a fat soluble vitamin along with other fat soluble vitamins which are vitamin D, vitamin A, E and vitamin K. So, today we will discuss only about vitamin A. So, there are different forms which are actively involved in the biochemical functions. So, there are 4 different forms of vitamin A. They are all related compounds, biologically related active molecules. So, in the form of retinol, retinol has alcohol attached to the side chain. So, this is the alcohol form of vitamin A, which is retinol. Retinal is the aldehyde form which has a aldehyde group in the structure. Retinoic acid has a carboxylic acid in its structure, and this beta carotene is the provitamin form of vitamin A which is found richly in plants. So, this beta carotene is made up of 2 molecules of retinols. So, these are the sources which richly contain vitamin A. So, you can see here dark colored leafy vegetables, green, orange, yellow colored vegetables are the rich sources of vitamin A. So, plant sources are dark colored leafy vegetables or green vegetables. Fruits and other plant sources, they have rich sources of vitamin A. Along with that, you can see some animal sources also. Liver, kidney, butter, egg yolk, they also have sufficient quantities of vitamin A. So, biological functions, vitamin A involved in lot of important functions. So, as retinal, as I said, the aldehyde form, vitamin A is very much essential for the vision. So, it is involved in vision. As retinoic acid, it helps in the growth and cellular differentiation. Also, retinoic acid is required to maintain the epithelial growth, maintenance of the epithelial cells of GIT, gonads, mucosal cells. And as alcoholic form, retinol, it is required for reproduction. And beta carotenes, as I mentioned, the plants which are rich in beta carotenes, they are having strong antioxidant action and they help in maintaining the immunity, or they are required for immune functions. So, vitamin A has these many important functions to be performed in the body in different forms. So, what is the requirement? This is the dietary requirement for vitamin A. For males, it is 1000 retinol equivalents, and for females, it is 800 retinol equivalents per day. So, RDA is mentioned per day. So, per day, this much requirement of vitamin A, or it has to be consumed. So, how this retinol and retinoic acids are functional? So, in our body, when we consume vitamin A in the form of either the plant source or the animal source, the retinol and retinal are the interconvertible forms. So, retinol and retinal can be interconvertible or interconverted by the enzyme retinal reductase using NADP as the coenzyme forms. So, it can get converted any time to retinal, or retinal can form any time to retinol. So, once this is converted into retinoic acid, this reaction, the second reaction which I am explaining now, is irreversible. So, whether it is retinol or retinal, once it is converted into retinoic acid, it becomes irreversible. That means, retinoic acid cannot form retinal or retinal. So, this is an irreversible reaction catalyzed by the system. So, once retinoic acid is formed, it cannot be converted back to either retinal or retinol. So, how this vitamin A can be absorbed? See, our diet has rich sources, or we have vitamin A richly found in either the animal sources or the plant sources. So, when we consume those compounds rich in vitamin A, they are absorbed from the intestine. Okay. So, in the intestine, the diet containing beta carotenes, the plant sources or the animal sources which contain the retinyl esters, are absorbed into the mucosal cells. So, beta carotene can be absorbed directly into the mucosal cell, whereas retinyl esters are converted into retinols by the release of fatty acids, and these retinol is absorbed into the mucosal cell. Now, this beta carotene, which contains two molecules of retinol, are released by the enzyme beta carotene dioxygenase, which requires bile salt for the conversion and the oxygen. So, in the presence of oxygen, beta carotene is converted into two molecules of retinol. And retinol, as I mentioned, retinol and retinol are interconvertible. So, retinol can be easily converted into retinol by the reductase enzyme, which is NADPH dependent. So, two molecules of retinol are formed, which is converted into the retinyl esters by the addition of fatty acids. So, fatty acids gets esterified to the retinol and retinyl esters are formed. Now, either in the form of beta carotene or in the form of retinyl esters, this vitamin A is getting incorporated into the chylomicrons, and we all know that chylomicrons are the lipoprotein, a type of lipoproteins which are formed in the intestinal mucosal cell after the incorporation of all the absorbed molecules. Now, this chylomicron is actually a lipoprotein which is required for the transport of lipids in the form of this. Now, this can transport also the vitamin A, which is incorporated into the chylomicrons. Now, in the form of chylomicrons, vitamin A gets transported in the circulation to various parts of the body. We will see how the chylomicrons take them to the liver, from liver how it goes into the different parts of the body. So, this is the absorption of the vitamin A through which we consume through the diet. So, once the chylomicrons are formed, the chylomicrons are released into the circulation, lymphatic circulation, and through the blood it will be taken to the tissues. Once in the peripheral tissue, chylomicron metabolism is over and the lipid particles are transported to the tissues, chylomicron remnant will, the carrying vitamin A will be taken to the liver, where the retinyl esters, whatever we have seen in the previous slide, the retinyl esters are stored in the liver. The chylomicron remnant carries it to the liver, and liver is the largest storage organ and it stores the vitamin A in the form of retinyl esters. Whenever it is required, retinyl palmitate or retinyl esters are converted into retinol by the release of fatty acids, and this retinol will be transported in the blood by binding to the protein called retinol binding protein. So, this is a protein that is synthesized by the liver, and that protein will carry this retinol. See, this is a fat soluble vitamin. So, it requires a protein to be transported in the blood and to take it to the various tissues. So, retinol binding protein carries the retinol which is released by the liver, and it will transport it to the various parts. So, the tissues where the retinol will be carried or transported and which has different functions are the eyes, in the retina or in the conjunctiva where it is required for the vision, then in gonads where it is required to maintain the epithelial tissues as well as for the reproductive function, in the skin to maintain the epithelial structure or the cell, the epithelial growth, and in the GIT, respiratory tract, genitourinary tract, where it is required for the maintenance of the cellular functions. So, this is about the transport, storage and utilization of the vitamin A. Next is how vitamin A mediates its action. The mechanism of vitamin A is similar to that of the steroid hormones, because we know that it is a fat soluble vitamin and its mechanism is similar to that of the cortisol, or the retinol and retinoic acid. The mechanism of action is similar, and retinol is a different mechanism that we will study a little later. So, retinol and retinoic acid behave like the steroid hormones, and it is similar to that of the cortisol. So, the receptors for retinoic acid and retinol are present in the nucleus. So, since it is a fat soluble vitamin, it can directly pass through the cell membrane and transported into the nucleus, where the receptors are specific, the specific receptors for vitamin A are present, and that receptors are called as the retinoic acid receptors in the nucleus. So, the vitamin A, that is the retinoic acid, is binding to the receptor in the nucleus. The activated receptor vitamin complex binds to the specific region on the DNA, which is called as hormone responsive element. Once the interaction between the receptor hormone complex and the HRE, that is hormone response element, happens, there will be alteration in the gene expression, that is mediated through the formation of the mRNA. So, we will see that here in this diagram. So, once the retinol binding protein transports the retinol in the circulation and it is taken to the cell, where retinol is converted into retinoic acid. So, the action of retinol and retinoic acid are similar to that of the steroid hormones. So, once retinoic acid is formed, as you know, this is a fat soluble vitamin, can easily pass through the cellular membrane and reaches the nucleus. Okay. So, in the nucleus, the receptor specific for vitamin A that is present, that is retinoic acid receptors, and the receptor vitamin A complex will go and bind to the specific region on the DNA. So, interaction of the DNA with the receptor hormone the receptor vitamin A complex alters the gene expression, or it induces the transcription. There will be formation of the mRNA. mRNA comes into the cytoplasm, and there will be the synthesis of the specific proteins, and there will be cellular differentiation, or the biological functions will be induced. For retinol, as I mentioned, retinol has a different mode of action. So, retinol is a component of visual pigments found in rods and cones. So, it acts through the Waltz visual cycle involved in the vision. So, I will explain this Waltz visual cycle in a while. Before we study about the Waltz visual cycle, we will know the relation between this retinol and the visual pigments. So, retinol is an integral component of the visual pigments present as rhodopsin both in rods and cones. The retinol present in the visual pigments in rods are responsible for dim light, helps in the dark, that is night vision, and those are present which in the cones are responsible for color visions. Now, retinol in association with the protein or the visual pigments in cones are of different types. Porphyropsin for the red colored vision, iodopsin for green colored vision, and the synopsin for blue colored vision. So, both rods and cones will have retinol in association with the visual pigments. So, rhodopsin in rods for the night vision, and retinol with the different types of proteins in the cones for color vision. So, these are the structures of rods and cones. Their membranes will have the rhodopsin with the opsin and the vitamin A as 11-cis retinol. In these visual pigments, 11-cis retinol is bound to the opsin and responsible for the vision. So, these visual pigments are found on the membranes of the rods and cones and responsible for both bright and color vision as well as in the night vision present in the rods. Rods for the night vision and cones for the colored or bright vision. Moving on to the Waltz visual cycle, the important biochemical function of vitamin A is its role in the vision. So, that will be explained with the help of Wald's visual cycle. So, as I mentioned, rhodopsin, which has the opsin plus 11-cis retinal, makes the rhodopsin, and rhodopsin is responsible for the dark light vision or the night vision. So, they are present in rods. So, once the opsin plus 11-cis retinal forms the rhodopsin, and this rhodopsin is responsible for the dark light vision or the night vision. So, the rhodopsin gets depleted, it is used by the body, and rhodopsin gets depleted. So, this rhodopsin needs to be regenerated. How that regeneration happens, we will see now. So, when the dim light falls on the rhodopsin, this rhodopsin breaks into again opsin plus all trans retinal. Okay. So, the conversion of this rhodopsin to all trans retinal and opsin is not a single step. There are series of the events that is happening, but this gets dissociated to opsin and all trans retinal. Once this all trans retinal is formed, inefficiently in the retina, all trans retinal can be converted back to 11-cis retinal, which can combine with opsin and the cycle gets completed. But this retinal conversion of all trans retinal to 11-cis retinal is inefficient by the retinal isomerase. That is why this all trans retinal is transported to the liver, okay, and in the liver retinal reductase converts all trans retinal to all trans retinol. See the difference, this is the aldehyde form which is converted into the alcohol form by the retinal reductase. This is a NADPH dependent conversion. All trans retinal is converted into all trans retinol in the liver. Now, this all trans retinal is converted to 11-cis retinol. Both are the alcohol form. By the retinol isomerase, all trans retinol is converted into 11-cis retinol. Now, this 11-cis retinol is converted to 11-cis retinal. Since this is a inefficient conversion, the all trans retinal goes into the liver and it is converted back to 11-cis retinal. Okay. So, this happens again during the dark. Okay. So, the regeneration of 11-cis retinal which can combine with opsin to form rhodopsin happens in the dark, and this is responsible for the visual cycle. So, this is a complete cycle in the dark, and when the light falls on this rhodopsin, there is photochemical changes happening and dissociation of opsin and all trans retina. Now, this again forms the 11-cis retinal by the body. Okay. So, this is the visual cycle, and because of inefficient conversion happening in the retina, all trans retinal will all the way to liver and it is converted back to 11-cis retinal. And what are the changes that happens in between? We will study that. The biochemical changes rhodopsin to all trans retinal is this. So, rhodopsin in the presence of dim light gets converted into bathorodopsin to lumirhodopsin, metarhodopsin 1, metarhodopsin 2, and finally it is dissociated into all trans retinal and opsin. So, it looks simple, but there is a series of interconversion of these rhodopsin to bathorodopsin, lumirhodopsin, metarhodopsin 1, metarhodopsin 2. Finally, it is dissociated into opsin and all trans retinal. And what are the biochemical changes happens? In the dim light, rhodopsin is changed. The previous slide we have seen all the intermediates that are formed during the conversion of rhodopsin to all trans retinal and opsin. So, a series of photochemical isomerization happens which causes conformational change in the opsin, leading to activation of the G protein, and the G protein is called as the transducin. Okay. So, G protein transducin causes activation of the phosphodiesterase enzyme. So, this phosphodiesterase converts the cyclic GMP into 5' GMP. So, there will be decrease in the cyclic GMP concentration, and this causes decrease in the sodium levels, and subsequently there will be hyperpolarization of the cell. So, this leads to the nerve impulse generated, and that is transmitted to the brain, which causes the dissociation of the rhodopsin to form opsin and the all-trans retinal. And we have seen how the all-trans retinal reaches the liver and it is converted back to 11-cis retinal. So, all that happens in the dark. Okay, so regeneration of the rhodopsin happens. So, this is the complete visual cycle with all the biochemical changes because of the dim light falling on the rhodopsin getting converted into all the in between forms, activation of the G protein, decrease in the sodium levels, hyperpolarization of the cells because of the activation of the phosphodiesterase, and decrease in the cyclic GMP levels. So, this is the mechanism of action mediated by the retinal through the visual cycle. So, this is the repetition again. This is to summarize what I have already explained it to you. In the dark, opsin combines with the 11-cis retinal to form rhodopsin and required for the visual cycle, and when the dim light falls on it, there will be conversion into all-trans retinal and opsin. So, this is the complete visual cycle and the regeneration of opsin and 11-cis retinal. So, moving on to the vitamin A deficiency. So, vitamin A deficiency can be studied into separate headings. It can be ocular deficiency and non-ocular deficiency, because vitamin A has a major role in vision, and that is related to the ocular functions, and other functions are separately studied as non-ocular functions. So, the deficiency can also be studied as ocular deficiency and non-ocular deficiencies. So, the causes for vitamin A deficiencies are mainly due to the decreased intake, obstructive jaundice which prevents the which affect the absorption. Then there will be liver cirrhosis and severe malnutrition. Why liver cirrhosis? Liver is the major organ which stores the vitamin A, and liver produces the proteins which help in the transport of vitamin A. So, any damage to the liver will affect the vitamin A metabolism. And severe malnutrition, again see, the proteins are required for the transport of these vitamins. So, severe malnutrition causes decrease in the synthesis of these retinol binding proteins, and nephrotic syndrome is a disorder where kidneys will excrete the proteins through the urine, or from the kidneys the proteins are excreted. So, these are some of the causes where vitamin A deficiencies are very common. So, how do we assess the vitamin A deficiency? Vitamin A deficiency can be assessed by dark adaptation time, where the dark adaptation time is nothing but the time taken for the person to visualize in the dark. So, that time will be increased in case of vitamin A, because the person cannot see in the dark. Okay, and decreased retinol binding protein concentration in the blood. So, sufficient protein, if it is not there in the circulation, vitamin A cannot be transported to the target tissues. So, by measuring the retinol binding protein in the blood will give you the assessment for vitamin A. Next is the decrease in the serum vitamin A levels. Normal serum vitamin A levels will be between 25 to 50 micrograms per deciliter. So, by measuring this, we can assess the vitamin A status of the body. Moving on to the symptoms, as I mentioned, the deficiencies are mostly closely related to their functions. So, we classify the deficiency symptoms into ocular and non-ocular. Even in the ocular symptoms, there are reversible and irreversible changes. So, ocular deficiency symptoms begin with the nyctalopia. So, nyctalopia is nothing but the night blindness. Okay. So, it begins with the effect, there will be effect of the synthesis of rhodopsin. So, as we have seen the visual cycle in complete, so the resynthesis of the rhodopsin is affected in case of the vitamin A deficiency. So, it is called as nyctalopia. So, dark adaptation time will be increased. And if nyctalopia, this is the beginning of vitamin A deficiency. If this can be easily corrected by sufficient supplementation of vitamin A. So, if nyctalopia is not corrected on time, it may lead to xerophthalmia. So, you can see this picture, xerophthalmia is because of the dryness and thickening of the conjunctiva. So, you can see here there will be lot of the membrane of the conjunctiva looks very dry, okay, dryness and increased thickness of the conjunctiva and cornea, leading to decreased lacrimal secretion and losing their opacity, and it become lusterless. So, again these are sequential progress of the disease. Once nyctalopia progresses, it leads to xerophthalmia. Again at this stage vitamin A deficiency can be reversed. So, these are called as reversible symptoms. So, by supplementing vitamin A in the diet, we can correct xerophthalmia as well as the nyctalopia. So, if nyctalopia and xerophthalmia are not treated on time, it leads to the Bitot spot. Bitot spots on the conjunctiva are nothing but greyish white triangular patches on the conjunctiva because of the thickness of the conjunctiva. So, you can see here greyish whitish triangular patches on the conjunctiva. Again, up till the formation of Bitot spot, the vitamin A deficiency can be corrected by the proper dietary supplements. Once Bitot spots produced on the conjunctiva, if it further proceeds, the deficiency proceeds, it may lead to irreversible changes. So, ocular irreversible changes lead to keratomalacia. You can see in this picture, there will be destruction of the cornea, cornea becomes soft and milky, there will be vascularization and opacity, the conjunctiva become opaque, okay, corneal ulceration will happen, and there will be infection of the cornea, and at this stage the person will lose the vision. So, if the nyctalopia, xerophthalmia and the Bitot spots are not corrected on time by the dietary supplementation, it will lead to a fourth symptom or the next sequential symptom called keratomalacia, and at this stage the symptoms becomes irreversible, finally leading to loss of vision. So, these are the ocular reversible and irreversible symptoms of vitamin A deficiency, and there are some non-ocular deficiency symptoms related to their non-ocular functions. They are follicular hyperkeratosis, where the skin becomes rough and keratinized resembling the goosebumps. You can see here, this is the hyperkeratosis of the skin, where there will be inflammation of the skin. Keratinization of the mucosal lining of the respiratory tract, GIT and urinary tract. Increased susceptibility to infections and cancer. Anemia and impaired immune function. As you know, vitamin A is required, it has a strong antioxidant action and it is required for the immune function. Because of the vitamin A deficiency, the person becomes susceptible for infection and the immune functions will also be altered. Moving on to hypervitaminosis, this is a small part where the overconsumption of vitamin A leads to hypervitaminosis. Why it is studied with vitamin A? Because vitamin A is a fat soluble vitamin and consumption of lot of vitamin A will lead to its deposition in various parts of the body. So, that may have certain other symptoms. So, hypervitaminosis A can be due to overconsumption of vitamin A more than 7.5 mg per day, leading to dry and pruritic skin, enlarged and cirrhotic liver, because vitamin A gets stored in the liver. So, liver becomes cirrhotic and it becomes enlarged. There will be increased intracranial pressure and congenital malformities in the fetus. So, overconsumption of vitamin A is also not good, that leads to hypervitaminosis. Moving on to the therapeutic applications of vitamin A, we have already studied the important functions of vitamin A, and vitamin A is required as the strong antioxidant immune functions, and it is required for the growth and differentiation of the cell functions. So, beta carotene, which has a strong antioxidant action, it decreases the cardiovascular diseases and required for this to prevent the skin and lung cancer. As retinoic acid, it is required for the epidermal functions or the growth and cellular differentiation. So, it is used for the dermatological problems in the form of the tretinoin, which has all trans retinoic acid, the vitamin A in the form of all trans retinoic acid, and it is used for topical application to treat psoriasis and acne, and in the form of isotretinoin, which has vitamin A in the form of 13-cis retinoic acid. It can be used as a oral supplementation, but it has teratogenic activity. So, this is used with caution. It is the tretinoin is used only for topical application for the treatment of acne and psoriasis. Overconsumption of the synthetic form of vitamin A through the oral supplementation in the form of isotretinoin can be teratogenic also. So, this is the therapeutic applications of vitamin A. So, I have come to the end of the topic. So, antagonistic to vitamin A, these are called as antivitamins which prevent the action of the vitamin A. So, they are the substances which make the vitamin A unavailable for its activity. We know the biological actions of vitamin A by consumption of these antivitamins or the antivitamin A. They prevent the availability of vitamin A for its biological activity. There are some compounds which can be which can be used as antivitamin A's. They are known as like mineral oils, cholestyramine and the olestra, which interfere in the vitamin A absorption. If the vitamin A absorption is affected, vitamin A cannot be transported to the different parts of the body where its action is required. So, the consumption of orlistat, a drug used in the weight reduction program, so that decreases the absorption of the beta carotene and vitamin E also. So, these are some of the compounds which can be used as antivitamins. So, that is all for today for this session. Thank you.